Okay, guys, a couple quick announcements as always to start off with. So I made these kind of announcements over during lab class yesterday as well, but we do have a Brightspace quiz today. So Brightspace quiz number 4, that's going to be, again, open until 2 a.m. tonight. Same format as the previous one. Make sure multiple choice, written response style questions, and yes, there will be some more Hardy-Weinberg style questions on that quiz then as well. The other things coming up then on Monday, we have the first exam. Remember that first exam, this one, most of my exams are going to be designed to be done in about 45 minutes to an hour for almost all the students. This first exam is a little bit of an exception to that, in that with the Punnett squares and the genetics, the Mendelian genetics, non-Mendelian genetics, Punnett square questions, with the Hardy-Weinberg questions, this one can take longer. So I will be, and I'm going to be in early for all the exams. We have an early morning class here. If you guys want to come in early for any of the exams throughout the semester to get started and get a little extra time on it, I'm going to do that. But I really heavily recommend that for this first exam, I'm going to be in here at 7:30 on Monday. And if you guys, I do recommend you guys come in early and get started early on that. I'm not going to stop you at 9:30 if you're still taking the exam and it comes around, but I would really prefer you to not miss any other classes. And this is the one exam that takes a little bit longer than some of the others. So I will be in at 7:30 on Monday for the exam. I do recommend that you get started early on it because it is a little bit of a longer exam compared to some of the others. Questions about that? Again, with the exam, it's going to be a mixture of multiple choice matching questions and some short answer or short essay questions. When I say that, it's usually a few sentences to a few paragraphs. Nothing too long. With those, though, it's probably going to be about 40% short answer, short essay, and about 60% of the points are multiple choice or matching questions. Tuesday next week, are going to have another pre-lab quiz that is due, but also remember you have a lab quiz also on Tuesday. And so with those lab quizzes, it's going to cover all the lab activities we've done so far, but that includes Mendelian, non-Mendelian genetics lab, where we did some of the practice problems in the human genetics material. It does include the evolution and natural selection lab then as well, and it includes the lab we just did yesterday that involved things like polymerase chain reaction and some other stuff. Okay. Questions about that? Lab quizzes are worth 15 points. All right. Otherwise, I sent out a small presentation. I said we got through, we didn't quite get through. I was aiming at the end of class on Monday to be through these last 2 sample Hardy-Weinberg problems. We didn't quite get through them. That being said, I did send out kind of a supplementary video on Monday to try to help you guys do the content worksheet questions. But I thought we'd go back through these real quick here as well. So I did send out a video to go through some of these problems to show how to do it. I'm trying to give you a lot of practice problems between the content worksheet and the quiz today to prep for the exam. But we'll go back through these real quick as well with Hardy-Weinberg, because Hardy-Weinberg is one of the things that does give students in the class a lot of trouble, for instance. And you can have it down one day, and then 2 days later, it's gone. So unless you're practicing it continually, it does tend to sometimes, you think you have it down and you almost do, and then you kind of forget a few things and 2 days later, and then it's getting confusing again. That's actually a completely normal thing to have happen then as well. All right, so with Hardy-Weinberg, the trick with Hardy-Weinberg is that I gave you 2 different equations. What were the equations? So P plus Q equals 1. That's one of the equations. But the thing is, P is a placeholder and Q is a placeholder. The thing you have to remember very clearly when you're doing these is what are they a placeholder for? What is P a placeholder for? What does it represent?
Frequency of one of these.
Yeah, so it's like the frequency of, say, allele number 1. Okay, maybe we say it's just a frequency of, say, a big A allele, just to give you guys a little bit more context. So what's Q then?
Frequency of
The frequency of the other allele. So if there's only 2 alleles at the gene locus, we'll just say that's little a, for instance, just to give you guys a little bit of context again. So these are allele frequencies. Now, when I'm talking about allele frequencies, it literally just means like the big A allele versus the little a allele. What percent of the population so maybe there are 50% of all the alleles are in the population are big A, 50% are little a. That would be your P's and your Q's. If you also ever hear me talking about gametes, those are the same thing as allele frequencies. Gametes are haploid. They only carry 1 copy of each gene, or they only carry 1 allele. So if, say, 25% of the gametes in a population is little a, that's another way of me just saying allele frequencies, because gametes are haploid. They're not diploid, so they cannot have a genotype. Does that make sense, everybody? Okay. Gametes can't have a genotype because remember, genotype in diploid organisms is a combination of 2 alleles. And gametes don't have 2 alleles for a gene locus because they're haploid. They only have 1 allele, 1 copy of the gene locus. Okay. So what's the other equation? What's the actual Hardy-Weinberg equilibrium equation then? P squared plus 2pq plus q squared equals 1. Now, here's the thing about these equations again. Context. You have to know that the different portions of this stand for different things. Sometimes you don't even need to use this entire equation. Sometimes you only need one part of it, okay, to solve some of the problems. P squared, though, the P squared portion, what is that a placeholder for? So it's a homozygous. And I usually say dominant, but it could be homozygous for allele number 1. It does not always going to be dominant, but it's a homozygous dominant genotype frequency. And what I mean by that is it might be the homozygous dominant genotype would be something like big A, just to give you guys again a little bit more context. Help place this one. What does the combination of 2 times P times Q stand for then? That's the heterozygous genotype frequency. But the heterozygous genotype frequency is all of that part together, 2 times P times Q. And yes, that P and the Q are the same ones as up in here. So if you know the values that P is a placeholder for, if you know the values that Q is a placeholder for, you plug them in. And then Q squared, that combination of Q squared, or Q times Q, is then the homozygous recessive genotype frequency. Okay. In essence, if this is a heterozygous, it'd be the big A, little a, and this would then be the little a, for instance, just to again give a little bit of context here. Now, the way to do these Hardy-Weinberg questions, with something like this one, I didn't say assume it's in Hardy-Weinberg equilibrium. That wasn't in there. In fact, is the population evolving or not was one of the questions. So you can't assume that Hardy-Weinberg is accurate. If you can't assume Hardy-Weinberg is accurate, you have to directly calculate allele frequencies. That's what we had to do on this one at the very end of class last lecture. But what I mean by that is you have to directly count the allele frequencies. You have to count how many big A's and how many, how many A's and how many B's there were in this one, and take the number of A's divided by the total number of alleles. Take the total number of B alleles, again, divided by the total number of alleles. So if I say, if I don't say assume Hardy-Weinberg is accurate, you have to directly calculate the allele frequencies, your P's and your Q's. Does that make sense, everybody? Here on this next problem, though, I said that after several generations, 9% of the animals displayed the recessive trait. Phenotypic descriptions are coded for by genotypes. So technically speaking, anytime I give you a phenotypic description, that's the same thing as also a genotypic description, because genotype typically controls phenotype. Okay. So if 9% of the animals display a suppressed recessive trait, the same percentage as the beginning of the breeding program. So is it changing over time? So is evolution occurring? So is it in Hardy-Weinberg equilibrium? Yes, because Hardy-Weinberg equilibrium describes a non-evolving, non-changing population. Not changing, not evolving means it is in Hardy-Weinberg equilibrium. That's one of the ways I can say it is in Hardy-Weinberg equilibrium. If it's in Hardy-Weinberg equilibrium, though, this equation will accurately describe the population and can be used to do some of the math involved. Okay. So in this case, when you're doing Hardy-Weinberg, you always have to start by identifying two things. The info that was given and the information asked for. So you always have to start off by clearly, when you're doing this, you have to identify what did I give you? And what I'm going to give you is some combination of 1, 2, 3, 4, or 5. There's 5 variables. I know there's 2PQ is this one, but we're going to put those and lump those always together. That's the heterozygous genotype frequency. In essence, I am always going to give you P or Q or P squared or Q squared. And actually, you can kind of ignore 2PQ as the information given to start. I'm almost always going to give you this, 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 or this in some form with some of these questions. In this case, which one did I give you? I gave you Q squared. So I said 9% of the animals display a recessive trait. But if they have the recessive trait, that means they have the recessive phenotype. And the only way they can have the recessive phenotype is if they are little a, little a. And again, that's the homozygous recessive genotype. And that number, 9%, is then the frequency of it. So what I gave you here in this case is Q squared. Okay. So I gave you Q squared, and Q squared together was equal to 9%, which is 0.09. So I gave you Q squared. And I also said that this does accurately describe the population then. I know, I was also already thinking I need to go over to the other side. Okay. So I gave you Q squared here in this problem. So 9% is the same thing as 0.09. What information did I ask for? So I asked for the heterozygous genotype frequency, right? So I asked for big A, little a, which is equal to 2PQ. Now, this is Q squared. 0.09 is Q squared, not Q. But how can I go from Q squared to Q? Q would be equal to the square root of Q squared. I just have to get rid of the square. You get rid of the square by taking the square root. Square root then of 0.09 is what? Point 0.3. All right, so that's my Q value. What's P? Remember, P plus Q equals 1. So if I sub in 0.3, I just said Q is 0.3. So we substitute in 0.3 equals 1, and then we solve algebraically. P is equal to 0.7. And then from that, I now know my value for P and Q. So I just have to substitute those in. Questions about that then? Okay, this one's a little harder. In a population of butterflies, the color blue is dominant over the color white. Blue is coded for by the big B allele, white is coded for by the little b allele. If 60% of the butterflies are blue, what is the frequency of the homozygous dominant individuals then? All right, so if we're doing this one, I said that 60% equals blue phenotype. Here's my question though. That again, phenotypic descriptions equal genotypes. But what genotypes would have then the blue phenotype? You would be big B, which stands for B squared is the variable that substitutes for, but also anything else? Yeah, plus the big B, little b genotype would also have the blue phenotype. So that would be the 2pq variable. And I said that those together were equal to 0.6. Now, can I solve for this? Why can't I solve for this? Can I solve for P or Q? No, because I have two different variables in here. I can't solve for P and Q simultaneously. I haven't got enough information from just this part of it. But is there a way I can solve for either P or Q? Well, what we just said is there's P squared plus 2pq plus Q squared equals 1, right? That's the Hardy-Weinberg equation. But I just said this portion right here is 0.6 plus Q squared equals 1. What that means, though, is what's Q squared equal? It means Q squared I can solve to 0.4. And then if I wanted to solve for Q, how do I solve for Q?
Big square.
Is again the square root of Q squared, which then was 0.4, which then equals to the square root of 0.4. 0.63. Okay, and if Q is equal to 0.63, what's P equal? 0.37. Because P plus Q has to add up to 1. And yeah, that's a little bit of rounding here. I usually round it to full decimal places for all of this. That's usually accurate enough for our purposes. Now, what did I ask for in this question, though?
Frequency of homozygous dominant.
I asked for the frequency of the homozygous dominant, which is what variable? What's squared? Frequency of the homozygous dominant genotype is p squared. And so this would be equal to 0.37 squared, which is what? Because 0.37 is p. So what you really need to do is have allele frequencies and genotype frequencies clearly understood. You know from reading these word problems, what did I give you and what am I asking you for? You got to basically say, did I give you a P or Q? Did I give you P squared, Q squared, or did I give you something funky like over here, but you can figure it out from the given information? Questions about that then? So I will probably have a mixture of probably a couple easy Hardy-Weinberg problems on the exam, but I probably also will have at least one harder Hardy-Weinberg problem on the exam that might be more similar to something like this, or the last couple on your content worksheet. Okay. Other questions then about Hardy-Weinberg. I want to make sure we really went through that because it is going back through it. Even if you watch the video, I know it was maybe a little boring to see me do it again here. But the idea here is that it is one of those things that does give the class a little bit more trouble. And if you're even interested, it's been on MCAT exams before. If you guys are pre-med students. Okay. Now, Hardy-Weinberg, again, it describes mathematically a non-evolving population. Now, obviously, in real populations, allele and genotype frequencies do typically change over time. Hardy-Weinberg is usually just a mathematical ideal that we compare against. We compare real actual observations from a field study against the Hardy-Weinberg mathematical description of the population of what you would expect in the absence of evolution. So usually when you're doing Hardy-Weinberg, you have observed data versus Hardy-Weinberg expected data. And that's what we're going to be doing in lab class next week as well. We're going to collect actual observed data, but then you're going to do Hardy-Weinberg expected data. We'll talk more about that on Tuesday, though, and fill in some of that stuff. Real populations usually do typically change over time. And there's 5 conditions that have to be met for allele and genotype frequencies to not change at all over time. And as we go through these, as you might expect, well, to meet all 5 of these conditions is something that pretty much almost never actually happens out in real world populations. Maybe for a brief few moments in time, they can do this, but typically some one of these is going to happen at some point on a population. So there's 5 conditions you have to meet to be in Hardy-Weinberg equilibrium. And these are the 5 conditions. You cannot have mutations. You have to have random mating. There cannot be natural selection at the gene locus. There has to be no gene flow. There has to be no individuals leaving the population or individuals entering new into the population. And there has to be extremely large population size, but in essence, this is a way of saying no genetic drift. So if any of these are not occurring, then it will result in change in the population over time. By the way, the one thing that people sometimes get messed up if I ask a question about these conditions on the exam is some of these are no, and some of these are yeses. And sometimes people get it mixed up. So just keep that in mind. Think about don't think about memorizing this necessarily, think about the context. And here's the context. We're going to go through each one of these and give some of that. All right, these are the 5 conditions to meet Hardy-Weinberg equilibrium or to not have change over time. You have to have no mutations. Now, obviously, that's just not something that ever really happens out in nature. There's always going to be a random background rate of mutation. And so, but sometimes the mutations could be at very, very low rates, so almost negligible. But if you're having mutations, then you're changing the allele composition of the population. Therefore, you're changing the allele frequencies. Again, point mutations, gene deletions, gene duplications, any type of mutations is theoretically creating new alleles, new variants. And if you're creating new alleles, well, then you're altering the allele frequencies in the population. So for instance, if you only had 2 alleles at a gene locus, and suddenly now there's a mutation that creates a 3rd allele at that gene locus, you change the allele frequencies. So my 2 mutations by itself change allele frequencies through the creation of new alleles. So if you have not to have no change whatsoever, you have to have no mutations. So again, you're going to have these creations. Sometimes you even have mutations that delete alleles also, such as gene deletions, for instance, then as well. So the creation, deletion of alleles naturally alters the frequencies and causes change in the population. All right, the other thing you need, another condition that you need to meet Hardy-Weinberg equilibrium is then random mating. Random mating just means that all of the alleles are going to be passed on equally to the next generation. But if you don't have random mating, if you have instead non-random mating, and by non-random mating, I mean preferential mate selection. If certain mates are preferred over others, certain males, certain females, if they breed more often, they pass on their genes and their alleles more often. This will decrease the random mixing of gametes in a population. Instead, their gametes, their alleles passed on at higher frequencies. This could be to a couple different things. One of the main reasons you get non-random mating is sexual selection. And again, we already talked about it, but this is mate choice. So for instance, peacocks that have male peacocks with brighter feathers, bigger feather displays are more likely to be chosen by a female little peacock here, and then they will mate. So any males with a bigger, more colorful displays will pass on their genes and alleles more readily to the next generation. That's non-random mating. And it causes change over time as a result of it. This is non-random mating, this is sexual selection. But even things like inbreeding, reproduction between close relatives, that can also cause then non-random mixing of gametes. So in certain small populations, especially in confined areas, you got various different animals, even sometimes plant populations that can get inbreeding, but that tends to then change allele frequencies over time. Even sometimes causes harmful alleles to become more prevalent in the population as well. That's actually one of the big problems with inbreeding, is it leads to a lack of genetic diversity. And sometimes it causes harmful alleles to become fixed into a population where they're the only ones present, for instance. Another condition to me, Hardy-Weinberg, is no natural selection. Again, if you're having natural selection, you get differences in survival and reproductive success. And this will alter allele frequencies. Again, natural selection is going to result in certain alleles, the more beneficial alleles, from being passed on to the next generation in greater and greater proportions over several generations. But again, that causes change in the population that would then be meaning that you're not in Hardy-Weinberg equilibrium. All right, and the process of natural selection results in an increase in those alleles that confer an advantage, a benefit, or an increase in fitness. In essence, natural selection is the cause again of adaptive evolution. Questions about that? All right, fourth condition then to meet Hardy-Weinberg is no gene flow. And gene flow, this is the moving of alleles into or out of a population. But as you move alleles in or out, that often alters allele frequencies and actually can alter allele frequencies pretty quickly. Typically, what I'm talking about with gene flow, though, is immigration. This is genes or alleles moving into a population from individuals moving into that population. Or it's emigration, which is then genes and alleles moving out of a population. So emigration is when somebody leaves an area. Yeah. With these, again, you can either have immigration or emigration. They are slightly different, but the end effect is pretty similar on populations. Gene flow. This is, again, where allele frequencies are altered through the movement of either fertile individuals or sometimes even just gametes. Just to make sure that you guys are understanding this, what I mean by, for instance, you can have pollen blowing on the wind from, say, one island to another island. Okay, so sometimes you can get movement of just the gametes, doesn't even require movement of entire individuals, but you can still have gene flow from something like that. But gene flow tends to then reduce the differences between nearby populations over time. So if you have one populations over here, one population over here, if you're getting some gene flow in between them, it tends to reduce their genetic differences over time. And this is because gene flow really can alter populations relatively quickly. Gene flow is actually more likely than mutation to alter allele frequencies. Immigration is, again, where you might have maybe an island out in the ocean from my examples here earlier. Maybe this goes from 2 green, 2 red, 2 yellow, 2 blue traits in my little population on the island, but maybe some individuals immigrate onto the island. So some individuals come out from some other population outside of the island and they move in. But now you've altered the allele frequencies so that the green is 3, blue is 3, yellow is 3, but now red's only 2. So what that does is it made the green, blue, and yellow traits more common in terms of their frequency, where the red trait now less common in terms of its frequency. So it altered the allele frequencies and the trait and variant composition of the population. Same thing can happen with emigration, but again, emigration is then when individuals move out of a population. Maybe they move someplace else, but that's still gene flow. But again, that alters it. If in this case I had a green, blue, and yellow individual move out, maybe that was the same guys that moved in over here. What ends up happening, though, is the resulting ending population. Now the red trait is actually increased in this population because it lost a green, blue, and yellow member. So that can alter the allele frequencies, especially when there's a lot of gene flow back and forth. That can create a lot of alteration of frequencies over time. Questions about that then? All right, last condition to meet Hardy-Weinberg is large population size. You have to have a large population size to restrict change in that population. The smaller a population is, the more likely that allele frequencies will fluctuate randomly from one generation to the next by genetic drift. Then, remember, genetic drift just describes random allele frequencies from one generation to the next. Now, genetic drift actually happens in both very large populations, but also small populations. But one random change in, say, a population of 1 million is not really going to impact the allele frequencies hardly at all. Does that make sense to everybody? But one random change in a population of, say, 10 or 100, that might have a 10 or 1% change. So think of the decline, my example I went through earlier was flipping a coin 10 times. Normally you expect it to be 5 and 5, but if you deviated just by 1, just flipping it, that's a 10 plus or minus 10% change. If you flip a coin 1,000 times and you deviated, you expect 500, 500, right? But if you deviated 1, you got 499 versus 501. That's only a plus or minus 1. And that's not, what's my percentage there? It's only like plus or minus like 0.1%. Does that make sense, everybody? So it's a much smaller. Once you get bigger and bigger, genetic drift is not really, doesn't cause as substantial of a change. But in small populations, genetic drift can substantially change them in a relatively short period of time. All right, again, genetic drift is always random allele frequency fluctuations, and the smaller a population, the greater chance of deviation. Also remember, and we talked about this already, genetic drift also can commonly cause allele loss. A lot of times drift can reduce genetic variation by the complete loss of some of the alleles in the population. Such as in my example right here, where you actually lost the white allele, for instance. Questions then about that? Questions about genetic drift again, because that is a concept that sometimes some students struggle with a little bit as well. All right, so again, these are the 5 conditions that populations would have to meet to be non-evolving and to be in Hardy-Weinberg equilibrium equation. And departure from any of these conditions was really what we just talked about, then will typically result in evolutionary change. Change, not necessarily adaptive change. Remember, adaptive change is always due to natural selection. But you can still have random change through genetic drift or some other factors, that random change through mutations, for instance. The other thing to keep in mind is populations can actually be simultaneously evolving at some gene loci while being in Hardy-Weinberg equilibrium at other gene loci. Gene loci, they evolve independently of one another. So you can be in Hardy-Weinberg equilibrium at one gene locus, but then they would actually be changing at others. All right, questions then about population genetics.
Could you give an example of a population that would be in Hardy-Weinberg equilibrium?
Probably no population would be in Hardy-Weinberg equilibrium for all gene loci. There's always going to be some change here and there. If you go at a particular snapshot in time and you get the right gene locus, you might see it being in Hardy-Weinberg equilibrium.
Would like bacteria or small organisms like that, would that be more likely than others?
No, because bacteria, something like Escherichia coli takes about 30 minutes to go through a generation. That means it can go through 30 minutes. It goes through like 24, it goes through almost between 40 to 50 generations a day. With something like E. coli reproducing that fast, remember, it gets a lot of mutations. It does a lot of DNA replication. DNA replication can result in mutations. So actually, probably microorganisms, bacteria, viruses are probably very quickly changing over time relative to us. Maybe not at a generational time frame for them, but compared to us, they evolve very quickly because they just reproduce so much, and that creates change.
There's somebody or like scientists trying to create viruses.
Yeah, there's a reason that Hardy Weinberg, I keep referring it to as a mathematical ideal. It's not something that probably actually truly exists out in nature. Like I said, you might get lucky and grab a population that for a certain time frame is in Hardy Weinberg, but it's probably not going to stick there over a much longer time frame. Hardy Weinberg is called, I call that a mathematical ideal because it's something to compare against. And yes, you can find some populations that are in Hardy Weinberg for a particular gene locus for a time period in some cases. But they're probably going to be not in Hardy Weinberg simultaneously at other gene loci. So no, I don't have a very good example. One of the few examples where, for instance, I can think of where there is probably, for instance, the beta-2 microglobulin gene is pretty much in Hardy Weinberg equilibrium for humans. But that's because beta-2 microglobulin, there's only one allele that exists for that particular gene in all humans. So it's what's called fixed. There's no variation there. And the function of that gene is so important that mutations are not tolerated, typically. So its function is highly conserved, and mutations typically have a loss of function mutation, and that actually results in your immune system not working. So beta-2 microglobulin is part of your immune system, is actually a really important part of your immune system. So that one doesn't show a lot of evolution over time in human populations. That would be one of the few examples I can think of off the top of my head. But again, it's a fixed gene locus of super important function. All right, and as always, I don't have like a separate study guide I sent out, but I have these what to know slides at the end of the presentation, which is basically my version of the study guide. But I'm giving it to you as we finish up lecture set, so you can even start the studying well in front of the exam. The what to know slides is not necessarily everything I might ask about on the exam, though. It's just the main things you need to know. All right, that is actually the cutoff also for exam 1 content. What did I just do? I don't know what I just, okay. Questions then over exam 1. Because otherwise I'm going to keep on going and start the next lecture set for exam 2 content. All right. So next thing we're going to talk about then is material from chapter 24 in your textbooks on speciation. So that first exam material is really covering genetics, population genetics, and evolution, and all those tie together. Again, Hardy Weinberg is used to assess is a population evolving or not. That's what population genetics often focuses on. Now we're going to, that's really microevolution. Now we're going to start taking a little bit more look at what is called macroevolution, how species are related to another, how species co-evolve together, how new species arise then as well. So speciation, we're going to talk a little bit about what is a species, but then we'll talk about mechanisms of speciation, such as allopatric speciation versus sympatric speciation. We'll talk a little bit about how do new species arise. And typically you require some type of either pre-zygotic barrier or post-zygotic barrier. We'll talk about what those is and how they operate on populations to produce new species or to keep species fully apart. And we'll also talk a little bit about how we classify species. This will then lead into here, our next lecture set's going to be on taxonomy. So then once we kind of define species and how species form, we'll talk about how species are related to one another and how we even determine some of their relatedness as well. So species. Species is a Latin word that means kind or appearance. The classical definition of a species. So this is the old school definition of a species, and I'll come back to this a little bit later here. But the classical definition of a species is a population whose members have the potential to interbreed in nature and produce viable, fertile offspring. So that's kind of the classical definition of a species. What does that classical definition of a species, though? What's it using? What process is being described here that's being done? Breeding. What kind of breeding or what kind of reproduction? Somebody said it. Sexual reproduction. So what this classical definition of a species hinges on is sexual reproduction. Two parents breed, sexually reproduce, and they make viable, that means living, healthy, fertile offspring that can then go off to reproduce themselves as well. Okay. So the idea here is if two organisms can mate, produce an offspring, and that offspring also can go off and produce kids like grandkids, they're considered members of the same species. Now that's kind of an important thing. So for instance, if you have a horse and a donkey, do you guys know what happens when you cross a horse and a donkey? Huh? What you cross horse and donkey, you get a mule. Okay. Mules are sterile every single time. They're actually a genetic dead end. So even though horses and donkeys can mate, they're considered separate species because their offspring is not fertile. And it's a genetic dead end as a result of that. Does that make sense, everybody? So you get in some weird situations along these lines. So this is one of those. Does anybody know what the other problem with this definition of the species really is, though? We'll come back to it. Okay, biologists nowadays, though, compare more than just breeding. Nowadays, they really are going to define species more on morphologies, physiology, biochemistry, DNA sequences. And they'll use a much more information to then see what a species is. Is that clock just really off? No, it's fine. Okay. All right. The traditional species definition of species, it is based on the ability to reproduce rather than on similarities. So, for instance, what are these birds? Do they pretty much look almost identical to you guys? Yeah, they're two different species. So this is one species of bird. This is a second different species of bird. I know they look, I can't tell the difference hardly at all between them. They look very similar. They have similar traits, but they are different species because they cannot mate successfully with one another. So these are two different, this is an example of similarity between different species. That being said, you also then have diversity within a species sometimes. So obviously I have an image here with a lot of diversity in different humans, but because they can still mate to produce offspring, they are all members of the same species. That makes sense. Okay. Typically then, and this is something we're going to talk about, this is the last thing for today, typically gene flow between populations is going to hold phenotypes and genetics of that population together. So as long as they're reproducing with one another, they're going to remain the same species. Okay. All right, so what we're going to, what's kind of definition of a species? What we're going to do is then talk about how now speciation, the process by which one species splits into two, we'll talk about that on Friday.